Last year, we focused on digital technologies. This year, we're focusing on innovations in different fields related to life sciences. When in late 2019, we decided the theme for this year's event, little we realized just how globally significant the topic would become in the months to come with the onset of the COVID-19 pandemic. While the field of life sciences has traditionally been one of the main drivers of research and development, certainly the pandemic has served as innovation catalyst in many different domains. It also highlighted how intrinsically connected public health and international security, national security can be when public health crises get really scaled up as the pandemic has shown. Over the next 2 days, you will be hearing from 20 world-leading experts, policymakers, innovators, entrepreneurs, and cutting-edge researchers from around the world. Together, we will be exploring how new technologies, new processes, and new actors are changing the ways in which biological agents are created, produced, and delivered. Experts will help us understand what kind of new opportunities and new risks are emerging in this field. And in particular, what are the implications of such innovations for international security, arms control, and disarmament? Now, before we start, one last remark from me. If you would like to ask questions to our experts, please feel free to do so at any point during each of the panels using the dedicated Q&A chat box. We will be monitoring the questions and pass them to the moderators who will then integrate it in the discussion. Now, without further ado, it is time for me to introduce our keynote speaker. Andrew Hessel is the president of Humane Genomics and the chairman of Genome Project Right. He's a scientist, he's an innovator and an entrepreneur, and we all really look forward to his brief. Andrew, over to you.
Thank you again for hosting this meeting and for inviting me. It's a pleasure to be speaking with you today. So my name is Andrew Hessel. I am a genetic scientist and cell biologist, currently based in San Francisco. I'm the founder of one of the founders and chairman of something called the Genome Project Right, and I'm also the president of a company called Humane Genomics that does virus engineering. And today I'm going to give you a little taste of my world and discuss some of the things happening at the forefront of life sciences. So I was one of these young people that was introduced to computers at an early age. I always loved the way things work. I took everything apart, and it was exciting. Mechanical and electrical things were terrific, but what really hooked me in was the personal computer. They were basically toys when I first started, but they were what really opened my eyes was here was a machine that by writing some code I could turn into other machines, and like many people of my generation, I was hooked. And back then, the way we stored programs and shared programs was using audio cassette tapes as the storage medium. So this was, it's quite old days. But as I started to refine my career, I moved away from mechanical, electrical, and computational systems more into living systems because I was fascinated by how does life work? And I was very bottom up. So I started working with cell biology and particularly in microbes. On the left here, you see a cartoon of a bacterial cell. On the right is a much more sophisticated eukaryotic cell, similar to our own cells. And what I started to recognize quite early on was the things that I knew about computers actually matched up to a lot of the things that I was learning about cell biology. And this is really exemplified in this particular diagram. We know that computers are built of electronic components. These make electronic logical circuits. These circuits are put together into chips and modules that put enough of those together and you end up with a freestanding computer. And that has some utility, but computers can be linked together to create networks. Whether it's a local network in your home or the internet, they can become much more powerful and sophisticated. And I saw a very similar hierarchy in biological systems, being made with proteins and genes, ultimately making biochemical reactions. More and more of those reactions come together to make metabolism. And with enough metabolism, you end up with a living cell. And cells themselves can be networked to form more complex structures, things like tissues and organs, and ultimately multicellular organisms. So this was a dream world for me because I really got to see these connections at a very low level very early on. Now, cells have machinery inside of them, subcellular components that are remarkable. Some of the molecular machines that are fundamental to all living things are the ability to copy DNA with DNA polymerases, make a working copy of DNA into a molecular cousin, RNase, which is kind of the working blueprints of the information, and then the ribosome, which you can think of as a protein printer that can decode the instructions on RNA and use that as a template for building both the structural materials and the catalytic materials of the living cell. This is totally universal and in all living things. And what I started to appreciate was that these molecular machines were like my computer, programmed with genetic code. Now, an E. coli bacterium has about 4.5 million bits of code, approximately the same amount of information as would be on a single file, a single photo on a phone today. Our much more complex cells, like our own cells, have gigabits of information, and they're organized, the chromosomes are organized in a much more sophisticated way, but they're essentially the same material. And that material is DNA. It's a molecule that encodes information. It's essentially a magnetic tape, very similar to what I was using on my early computers. So this molecular tape is not digital. Well, it's digital information, but it's encoded in atoms. And for me, if I loved computers at the beginning, I absolutely adored working in cell biology and genetics because it was just, I could just see the potential for programming living things. Now, the biggest difference between computers and biology is manufacturing. On the left here is an early assembly line for Mac computers in Silicon Valley. You can see they're being assembled by hand today. They're usually done by robots. But on the right is the molecular self-assembly and reproduction of bacterial cells in time lapse. And this is truly the most advanced form of manufacturing in the world today. It's taking elemental materials and making complex molecules and ultimately more living cells. In some ways, it's printers that make more printers. So the types of things that I really try and teach people today is that DNA is a digital programming language that directs all cellular form and function. And it's truly digital code, but it's not electronic bits. It's physical atoms. And the information is executed by universal molecular machinery. So we have a universal code running universal molecular machinery, and this gives rise to all the diversity that we see in nature. In other words, nature is generated by software, not so much hardware. Today, we know that the world is undergoing unprecedented technological, economic, and social change because of digitization. It's touched all of our lives. But probably the most powerful thing happening in the world today is that life itself is being digitized. I started to see this process unfold early in my career with the start of the Human Genome Project. To me, this is as important as the Apollo projects and the moonshots. It took 13 years from start to completion, and its entire purpose was just to read the blueprint of the human form, of our bodies. And along the way, it unlocked processes, tools, and technologies that really allowed us to go into and explore biology in ways we had never done before. It brought automation to genomics. These are high throughput sequencers that were installed at Celera and used by other groups as well, that started to make the process of reading DNA code pretty much automatic and run by technicians. And this continues to this day. This is an image from BGI, a sequencing powerhouse based in Shenzhen. You can see it's automated machines. They're just higher performance. Each machine here can do human genomes in a matter of hours. It also catalyzed one of the most remarkable technological advances. We saw the acceleration of sequencing technology at a pace that was never before seen. It outstripped Moore's law by up to 500%. And today we can sequence megabases, millions of bases of DNA for just pennies. And it's still accelerating. The overall process of sequencing starts with an organism, whether it's a human, a plant, a microbe. And if it's not a single-celled organism, we reduce it to single cells, we pull out the genetic material, we run it through these automated sequencers, and ultimately, at the end of the day, we end up with a file on a computer system. This has been done for about 55,000 different organisms today. And of course, millions of people, what you can think of as instances of the same organism, have been sequenced. This really, by sequencing, we get down to the very code level of life. So we get an understanding of life at really the lowest level possible, how it not only runs molecular machinery, but how it builds it. And today we've organized that information in some pretty remarkable databases. This is the Kyoto Encyclopedia of Genes and Genomes. And this looks like a static image, but it's actually a live interactive database showing essentially the wiring diagram of a single cell. It's complicated, but we can drill down on any particular part of this metabolism and start to see the exact biochemical pathways that are being catalyzed. The numbers that you see here in the little boxes correspond to enzymes, and if I were to click on those, I would be able to see the protein sequence of that enzyme and ultimately the genetic code that encodes it. So this is truly the roadmap and the blueprint, the program of a living cell. What has been central to my life professionally is that we can also write and edit this DNA code. This isn't new. We've been doing it for over 40 years, but in the early days, we really had to start with the physical material of DNA, special enzymes that worked as scissors to cut the DNA in precise points, and other enzymes that acted like glue to stitch it all together. And the result is something we call gene splicing or recombinant DNA technology. Now, I did this quote a few years ago where I sat down with a stack of magazines and scissors and glue and wrote out this single sentence. It's a quote by Stanford synthetic biologist Drew Endy. And just writing this sentence took me about an hour and a half of work. To write a small bit of genetic code using these types of technologies meant that you had to work with a molecule. that you cannot see. So this required extensive training, the proper facilities, access to these particular enzymes, and validation technologies to actually read the code that was being written. Clearly, this was difficult work to do, but it was good enough, sufficient, that we were able to make the first proteins and really found the biotech industry. Human insulin being one of the first proteins we ever made, and radically changing the life for diabetics around the world. Previously, insulin had to be harvested from organs. Today, this type of editing and manipulation has all been digitized. We use software tools rather than very similar to a word processor to do the editing and splicing. It's trivial now to search for a string of genetic letters, to recombine, to move things around, and all the annotation is clearly visible in the code. This is a big step. But now to go from this digital world back into the physical, we need some way to make the molecule. This is done using a process called DNA synthesis. And today, DNA synthesis looks a lot like the printers you might have on your desk for your word processor. This is a device that is a desktop unit for the lab. It can string together up to 7,000 bits of genetic code, sufficient to build almost any protein. It can be used to reprogram metabolism and even make small viral genomes. It's quite a sophisticated device. But just as in the early days of printing, if you needed to do a bigger print job, you had to go to a service. And today, most of the DNA that's being written is being done by services like Twist Biosciences and others that do high performance, high throughput DNA synthesis and assembly. These are truly DNA molecule printers, and they're getting more and more powerful. But it's a key step in the digitization, and it's really the foundation of the technology of synthetic biology. Groups started to use this technology from essentially their birth. One of the first companies to do this was Ginkgo Bioworks. Ginkgo Bioworks was founded out of a group at MIT, still operates out of Cambridge. They call themselves the organism company because they are literally using synthetic DNA to reprogram the metabolism of cells to make important compounds. They've grown extremely quickly. They're just over 11 years old. And I want to be clear, when I say that you can, when you write a genome, what I'm actually saying is you're building the organism, because as I showed earlier, the genetic code programs the activity, the form and function of the cell. So if you can write a complete genome, you can build organisms from scratch. And this is profound. We're moving well beyond just synthesizing a protein or reprogramming a little bit of metabolism. We're actually starting to be able to write complete genomes as these technologies have advanced. The very first genome that was written was only done 18 years ago by this researcher, Dr. Eckhard Wimmer at Stony Brook University. He wrote the genome of a virus called poliovirus, about 7,500 bits of code. It was a real breakthrough at the time. It took 2 years to write that genome and over $500,000 of chemical DNA synthesis. But it demonstrated that we can build organisms from scratch and that they are identical to the natural organism that had been sequenced and deposited in the database. Today, this technology is being used in many ways. My company, Humane Genomics, for example, uses it to build viruses that are custom designed to hunt down and kill cancer cells. The virus that we use is called VSV, and it is literally the shape of a bullet. So I like to think that we are programming magic bullets to kill cancer. And as we go around the cycle of designing this virus, building the virus, and ultimately testing it on cancer cells, we get faster and better and cheaper. And today we can build custom viruses in just a few days for almost any cancer using these digital tools. But similarly, the same technology was used just in the earlier this year to reconstruct the SARS-CoV-2 virus after the Chinese researchers had sequenced it and put it into the public domain in early January. It took less than a month to make the virus, which allowed it essentially to be recreated in a lab in Switzerland, as well as to start the development of vaccines and diagnostics very, very quickly at a rate we've never been able to do before pre-synthetic technologies. We've even gone to the point now where we can synthesize whole genomes for single-celled organisms. And the great ambassador of biology is E. coli, bacterial cells. That genome was just synthesized last year, about 4 million bits of code. The entire process took about a year and a half. But now that it's been done, it's a template for building other bacterial cells, and the process would go much faster. And also yeast, which is kind of the foundation of modern biotechnology and agriculture, has been synthesized. This is significant because the yeast genome is about 2.5 times the size of E. coli. It took an international team many years to do it. There were many technical problems that had to be solved because the yeast chromosomes are actually much more similar to human chromosomes than they are to bacteria. So this is the first eukaryotic cell to be synthesized. So clearly it's early days in being able to design and build cells, but we can see where the trends are going. Routine work with current DNA synthesis capabilities has already unlocked the ability to write any protein, from antibodies to enzymes, and do it in a way that is highly precise and scalable. Within reach, we can now essentially theoretically make any virus from scratch, opening the door to new vaccines and gene therapies that would have been very difficult to make in the past. We can reprogram the metabolism of almost any cell by adding mini chromosomes or adding to existing chromosomes new features. And with edge technologies, we're now moving into single-celled organisms. But as we start to move into larger and larger constructs, we're going to be able to make larger chromosomes in the 100 million base pair range and larger. And ultimately, assembling multiple chromosomes opens the door to making any natural animal or plant. So we're clearly starting to unlock the capability of designing and building the organisms that we're used to on the planet with these technologies. Essentially, we're entering an era where organisms can be programmed as easily or as hard, depending on how you see it, as today we program computers. But why do this? Well, genome engineering allows really precise control over an organism's form and function. It teaches us how life works. It allows us to truly understand the operations. It accelerates the development of applications ranging from novel therapeutics to vaccines and therapies. And it opens the door to completely novel capabilities because when you work at the code level, there's no species barrier. So this technology allows us to actually enhance the capabilities of nature by being able to mix and match features from the natural world across distantly separated species. In many ways, it's truly a revolution in evolution. And this is profound. I don't mean I realize that this scares people in the same ways that in the early days of robots and computers, many fears were articulated. To teach people about these technologies, to explore the technology, the science, the ethics, and help create the standards and guide society forward. a group of scientists got together in 2016 and formed an organization called the Genome Project Right. This is really the sister organization of the Human Genome Project. Only this time it's not about reading the genome, it's about being able to write the genome. So it's really a genome project for a completely new generation of researchers that now aspires to be engineers as well as scientists. This starts to close the loop of organism, file, organism. Again, we've sequenced about 55,000 genomes. We've only synthesized about 50 organisms. The vast majority of those, about 47 being viruses, which have very small genomes, and only about 3 cellular genomes. So there's an imbalance in this cycle, which I tend to think of as the synthetic genomic cycle, but it will start to close. Just being able to go around this cycle gives us new powers in conservation, because now nothing has to go extinct. We can teleport or transport, or you can think of it as faxing organisms from one location to another, as long as we can send a file. And we can even de-extinct recently extinct organisms that we've sequenced already. So this is a very powerful technology. Again, very early days, but and I want to be clear, this is not genetic engineering in the classical sense. It's just digitizing and then going back to the original organism. No modifications need be made. But if you do make modifications, you start to branch out of this cycle and you start making engineered organisms based on natural templates. And this is a vast area to be explored that we've only just scratched the surface. So what can we expect with this technology, the synthetic biology cycle, as it continues to advance? Well, we're going to see, number one, more access to DNA sequencing and advancement of that technology and the analysis of that data. Even though DNA sequencing is relatively standard, it's still not easy to access for most people. But clearly, like computers, it will be democratized. DNA synthesis technologies will equalize with reading technologies. So read-write will balance out. And this is key, because again, we've got 55,000 organisms sequenced, only 50 synthesized. As we can go around that loop, it will give us incredible new capabilities, but the price performance will match very quickly. This should be, since a human genome can be read today for under $1,000, just imagine being able to synthesize a human genome for $1,000. This will be possible within the next decade or two. We're definitely going to need and can expect better computer-aided design tools to be able to design organisms and engineer them, because today they're not that powerful or sophisticated. They're mainly editors. They will become much more AI. And of course, I've shown you various devices that either sit on a desktop or fill a room. We can expect that this automation will advance and be miniaturized and standardized so that our labs will start to be more and more portable and powerful, particularly in the areas of test and measurement. And of course, this technology, as with all other digital technologies, will give rise to new companies with new capabilities, making new applications, some of them impossible to consider before, and really fuel the growth of the bioeconomy, which today is approximately 2 to 3% of GDP. So it's really, this will create and fuel profound changes in the world. And just as some of our digital companies have started quite small, in this home in Silicon Valley, 2 people got together and created a little company called Apple Computer out of that garage. in less than 45 years, that company has become the most valuable company in the United States as of yesterday, with a market capitalization of over $2 trillion. We're going to see companies starting out of a small lab with relatively unsophisticated equipment turn more automated, like this lab, the Emerald Cloud Lab, or some of the newer, more modular cloud labs like Zymergen, a company that does specialty materials. We'll start to see it advance and these companies become much more powerful. And of course, these capabilities will be available to more and more researchers in fields, not just in life science, but in orthogonal fields and complementary fields of engineering. How do people, countries, companies harness these benefits? Well, number one, you have to commit to them. Even if there's not full consensus that they should be explored. It's pretty clear to me that genetic engineering has powerful benefits. And even though there's sometimes pushback in their exploration, there needs to be a commitment to move forward. A strategy and vision has to be created and implemented. This is absolutely key. Otherwise, there's no coordination. Standards need to be developed for these tools, for access, for digitization, and for automation. And particularly key for me is that education needs to be comprehensive, starting at the earliest ages. It surprises me today that many kids know more about computers than they do about the technologies that make the living world and themselves. I've been training my own daughter, who's in kindergarten, in these technologies, and I know that they pick them up very, very quickly, just as they pick up computer technologies. It takes investment, significant amounts of money being invested in biological science, engineering, biomedicine, and of course, entrepreneurship, because we have to start translating these technologies into applications that people can use. And importantly, and a big role for government in particular, is to consider regulatory environments and wherever possible, remove barriers, make sensible regulation to advance these technologies in a safe and responsible way and allow entry of new products into the marketplace and really coordinate activity. But what are the risks? Today, I think one of the largest risks is that this work is simply not as transparent as we'd like it to be. Most labs exist as almost single cells, not working as a complete whole. It's difficult to understand the work that's being done. It's hard to do oversight on a continuous basis. And there's very little international coordination. And we've seen some of that just in the recent COVID outbreak. Lots of activity, not that much coordination. This is a missed opportunity. There's tremendous asymmetry, just as in computing, between things that can be made today and securing systems from those things. And I'm thinking specifically along the lines of things like computer viruses, which are relatively easy to write and yet requires very sophisticated systems to defend against. I think we have some tremendous challenges with small genomes of viruses. And I think, if anything, our recent experiences are calling to light that we need to rethink virology from the ground up to make it more transparent, to have the appropriate biosecurity systems and sensors in place. There's always the risk that technologies can be used inappropriately for warfare or terrorism, or simply by making honest mistakes that were unpredicted. I think with biology, we must be very careful here not to go down the path of using biology as a weapon. It's an incredibly powerful technology. I look at it as not atomic weapons, but atomic scale weapons, because these are really nanomachines. Of course, there's the risk of societal resistance is large, and that there will be an immune response to the development of these technologies because of lack of trust, lack of understanding, or other issues that where people just don't accept them. And I think that together, the real risk for many societies and perhaps for humanity is just missing the boat, not taking action and not trying new things. This is always the weighing the risks and benefit of any new technology. But for example, it's even with the development of COVID vaccine, it's clear that we have to be able to respond faster if we're going to protect society from viral outbreaks. And we've had plenty of opportunity to take these actions and earlier. For example, we still haven't conquered the flu, even though we accept globally, many hundreds of thousands of deaths each year. In closing, I want to just point out that this technology, like computer technologies, is probably unstoppable because it is so attractive to young people. It's powerful. It can do many, many positive things. I think the positives greatly outweigh the negatives. And young people naturally gravitate to this type of work. This is a photograph of the International Genetically Engineered Machines competition, what you might think of as the Olympic Games of synthetic biology. It's been training students now since 2004, and it has become the premier program for training young people in these technologies, teaching them about the social practices, ethical implications, safety engineering, and more. And all I can say is if you go into a room filled with 3,000 or 4,000 young people that have been working collaboratively on these types of projects and feel the energy of their excitement and the incredible technologies that they're working on, you realize that this is truly a force that's going to just keep growing and growing this century. I truly believe that our ability to understand and engineer life will be what defines this century, and it will be young people that lead us forward. So with that, thank you for listening. Please feel free to reach out with any questions. I'm happy to point you to source materials or other examples. Thank you again for allowing me to speak. I really appreciate it.
Thank you, Andrew. This was a great start for our innovations dialogue. You covered basically every single aspect that we're going to try to elaborate on over the next few days. And you touched upon pretty much everything, from the way in which technology has evolved over the last few decades, but also looking forward, where is it going and how is it going to change? And I understand that because of the advances in digital technologies, in computing power, etc., the pace of innovation in the field of life science is going to potentially increase exponentially, so compared to what it was before. In one of your last slides, well, you had two slides about benefits and about risks, and I had one question that was actually also coming from one of our guests in the audience about the regulatory framework and how hard it is to strike the right balance between having not too much regulation to allow innovation to kind of emerge and take place, but enough regulation to avoid any of the unintended consequences that you have listed in your risks. Particularly if I reflect, keeping in mind our audience, the risk of biological warfare, biological terrorism. And you've basically touched on the fact that these new technologies and the way we're going are going to democratize access to these kind of technologies at a much lower price range and a much faster kind of turnaround time. So what do you think is the role for international regulatory frameworks? Traditional frameworks, we have international treaties like the Biological Weapons Convention. Is it really fit for purpose, or is there a role for other actors that are not necessarily state actors to come together? What is the role of industry in making sure that we can harness the benefits, but not forgetting that there are also concrete risks on the other hand?
That's a great question. I don't have all the answers, but where I tend to look for examples today is not so much in the field of life science, but the field of computing and robotics. Because these are, in general, computing and robotics have advanced faster than education or regulation. It's really the challenges that come up and the lessons that are learned on the fly. But one of the positive aspects of building a digital world is that we have a better understanding. First of all, it's a virtual digital world for the most part, and we understand many of the dynamics that happen in that virtual digital world. Basically, if a technology exists and it has a weakness, it will be exploited. There will always be some exploits, but the number of people exploiting a technology for harm is small relative to the people that will work to secure it. So this is why, even though computers are arguably less secure than biology, they operate and still run our world today reasonably well. There are areas where we're not so sure that we want computers to work, like in essentially the management and decision-making around nuclear weapons, because they're a hack on a system could potentially lead to thermonuclear war, something that couldn't be stopped. But in many ways, we've learned some of the things that need to be done to secure digital foundations. With biology, most of it is digital until you hit print. So that moving from that virtual design and engineering world where you can simulate and run models, that's a contained world. You can analyze it. And part of the problems that we have today is we can't fully simulate even a single cell, but we will get there. And it will probably be necessary to do better and better simulations of biological systems virtually before we commit to a print. And once it's printed, of course, it's in the real world. There, we need to make sure that it's highly secured. I am actually becoming a very strong advocate of hard separations between engineered biology and natural biology, for the simple reason that we don't fully understand the biological systems in our natural world. But I come from a background of drug development where just about everything is done in a bioreactor and is fully self-contained, and it's never supposed to, drugs don't just leak out of pharmaceutical companies. I think that as for synthetic biology to flourish, we're going to need transparency in the design and engineering process, transparency and regulation in the print processes. I think that needs to be continuous in real time. And once things are printed, the test and measurement and containment needs to be much needs to be much better and more secure than it is today. So we need to have essentially stepwise controlled release, and this needs to be rationalized at a global level. Because if it's not, then it will just be it'll be an uneven playing field. And of course, as we've learned, there are no borders to biology. So this is an opportunity to create new frameworks that are digital, real-time, and new systems for management that will allow this field to flourish.
Thank you, Andrew, for that. And I'm afraid we've run out of time. We're already 6 minutes after the scheduled time, but it was such an interesting conversation, and I wish we could just carry on, and hopefully there will be other opportunities. I also appreciate you connecting so early, considering that you're based on the West Coast in the US. It's very early in the day for you. We hope you have the chance to stay with us and see the rest of the conference. If not, it will be made available. And thank you again for joining us. There will be a short break now, and after the break at 2:30, we will start with our first expert panel. This session will give participants, all of you, an introduction to gene editing technologies, particularly CRISPR, and explore the risks and benefits that this technology can bring.
Excellencies, ladies and gentlemen, it's a pleasure for me to moderate this opening panel, and I would like to thank UNIDIR for its excellent leadership at the convergence of security and emerging technologies. My name is Eleanor Powells. I'm a senior fellow with the Global Center on Cooperative Security in New York, and I spent the last decade working closely with governments, the UN, and private sector actors on the convergence of AI with modern biotechnologies. Our panel today will open with a presentation by Professor Pawan Dar, who is Professor and Dean at the School of Biotechnology at GNU. Professor Dar will help us understand how genome editing technologies operate in the biological substrate, what latest breakthroughs scientists have achieved, and the types of applications we see emerging in functional genomics. Our second panelist will be Piers Millett, Vice President of the IGEM Foundation and Senior Research Fellow at the Future of Humanity Institute at the University of Oxford. Piers will give us exposure to the global community of young practitioners involved in genome editing through IGEM. So he will demonstrate the role that such an international competition plays in framing the governance and dual-use challenges we face in genome editing and gene synthesis. After our two presentations, we'll have a moderated discussion, including questions from the audience. So let me remind all of you that from the beginning of the panel, we welcome your questions through the Q&A chat box available on WebEx. Our discussion today is taking place at a defining moment for the life sciences. Humankind is facing a public health crisis unprecedented in magnitude and global scope. Scientists, but also member states and citizens, are faced with renewed complex questions, hopes and concerns about how to govern the opportunities and risks coming from life sciences. And going forward, one of the most significant legacies of this crisis will be the way that the pandemic dovetails with another major global disruption, the convergence and democratization of dual-use biotechnologies. Among these powerful technologies, genome editing provides us with new exceptional capacities for making precise additions, deletions, and alterations to specific DNA segments, promising to drastically improve human health. Beyond medical advances, genome editing is also an epistemic revolution as much as a technological one. So the capacity to precisely edit specific DNA segments within living cells provides new powerful means to test and predict genomic functions within the biological substrate. Even more, the convergence of genome editing with artificial intelligence could improve functional genomics to the extent of being able to assess how genetic sequences are meant to function before being assembled, and even if the combination is not seen in nature. Therefore, we are not only discussing a set of technologies and bioinformatic tools, but a new form of tacit knowledge, with dual use potential that we need to govern and possibly control. And indeed, genome editing is also a landmark development for the field of international security and arms control. And today, we aim to have a balanced discussion about opportunities and risks. For instance, we want to avoid hyped scenarios, but scientifically assess whether genome editing technologies are a game changer in the conceptualization, design, and production of biological weapons. Are we able to define, understand, and prepare for a taxonomy of potential threats? For instance, around the use of genome editing to augment the capacity of pathogens to infect a host, acquire multi-drug resistance, or evade the immune system? What about potential threats that could target non-human genomics or essential functions of our bioindustry? How well prepared is the international policy community to identify, screen, and mitigate emerging biological threats? Biosecurity controls that rely primarily on controlling access to dangerous existing pathogens may be insufficient. So how can we learn to increasingly rely on functional genomics approaches, AI and bioinformatics, to better emphasize genetic functions and better assess the risk of unknown or modified DNA sequences? So without waiting any longer, Piers, the floor is yours.
Thank you very much. So it's always nice that you've seen a picture like this before. One of the final slides that Andrew showed you was a very similar photograph of IGEM. I'm going to tell you three things with you today. I'm going to tell you a little bit about IGEM, I'm going to tell you some details of our safety and security program, and then identify some specific examples of biosecurity lessons that we've learned over the last decade. IGEM, the IGEM competition is an annual worldwide synthetic biology event aimed at undergraduate university students, as well as high school and graduate students. Multidisciplinary teams work all summer long to build genetically engineered systems using standard biological parts that we call biobricks. IGEM teams are local people working to solve local problems with a global impact thanks to the power of synthetic biology. Unsurprisingly, for a biological engineering competition, many of the competitors and participants are biologists and engineers. But teams often have students from a very diverse range of fields, for example, computer science, art and design, physics, marketing, law, mathematics, as well as the social sciences. IGEM is also genuinely international. Last year, it brought together over 6,500 participants in almost 350 teams from over 40 countries, every inhabited continent of the world. To date, it's produced somewhere between 40 and 45,000 alumni. That's 40 to 45,000 champions for synthetic biology that have gone on to have academic careers or found companies, or in some cases come back and work for things like IGEM itself. But IGEM is much more than a science and engineering competition. It has a real impact in the real world. This is a press release for a $35 million deal to develop a new therapy for celiac disease. This is a screenshot from the webpage of the company. It is a press release that it makes clear that this new therapy has spun straight out of an IGEM project. In fact, it spun out of this IGEM project. Back in 2011, this team were engineering bacteria to break down gluten, and that was translated into a new therapy to break down gluten in the gut and therefore treat celiac disease. And here is Ingrid Swanson-Pultz, the leader of the IGEM team that did that project and now Chief Scientific Officer of PVP Biologics, talking about their clinical trials at the Giant Jamboree in IGEM a couple of years ago. This is just to stress that members of our community have gone on to raise a considerable amount of money, found companies, and that therefore IGEM really is a talent pipeline for a notable part of the synthetic biology industry. But we believe that good biological engineering is more than just good technical work. Since its inception, IGEM has embraced what we call human practices. All of our teams have to think about how their project affects the world and how the world affects their project. The teams are encouraged to look at different facets of their work. And as you can see on this slide, safety and security is a core part of what we believe makes up good synthetic biology. Our safety and security program is built around safe and secure project design, safe and secure lab work, and safe and secure delivery, both in terms of physical material, but also in terms of information. IGEM believes that safety and security is everybody's responsibility. We have expectations for all of the students, we have expectations for the instructors and PIs, and we have expectations for IGEM itself. Together, they ensure that teams meet international, regional, national, and institutional rules and regulations. And we expect teams to look at safety and security issues throughout their project, in particular, telling us about it when they're moving from the design to lab phase, and again, from the lab phase to communicating their work. All teams have to provide us basic information on what they're doing, any hazards associated with that work, and how they're managing the risks that those hazards may pose. And then any team that wants to work with anything that's a little bit more dangerous has to provide additional information so that we know that they understand the risks involved and that they're managing them appropriately. At the heart of that, IGEM maintains a whitelist. It tells teams what they can do without having to check in greater detail. That is roughly equivalent to what you would expect to see in an undergraduate biotech laboratory in any university anywhere in the world. That list also has examples of things that may be a little bit more hazardous. That's organisms, parts, and activities that they have to come back and give us more details on. There are also some things that can't happen inside the competition that we don't allow. For example, teams are not allowed to work in high or maximum containment laboratories, and therefore can't use anything that would require that sort of biosafety levels. At the heart of our work is a safety and security committee that has members at the moment from North America, South America, Europe, and Asia. We're always looking for a wider diversity of members, but the members of this committee have a wide variety of different backgrounds. We have current and former regulators from different parts of the world. We've got laboratory biosafety professionals, contained use and environmental experts, biosecurity and dual use specialists, bioethicists, animal use experts, so a wide variety of skills to draw upon. All of that information provided by teams is screened by an external partner. They are looking to see whether the teams have identified all of the hazards from their work and whether the measures that they've put in place do manage those risks appropriately. Anything that they spot that may not be entirely as expected is then referred back to that safety and security committee I just introduced to you. All of the parts, the biological parts used in the competition, are also screened by a different commercial partner. The vast majority of parts used in the competition pose minimal risk. There are some parts that may pose some hazard if used in certain ways in combination with other parts or for specific purposes. Those have a safety flag on and you need extra permission to use them in the competition. There are no dangerous parts in our registry, for example, human toxins. You can think about synthetic biology and biological engineering as an attempt to make a genuine engineering discipline for biology. I know we've talked about genetic engineering in the past, but this really isn't an engineering discipline. At the heart of engineering is this design, build, test, learn cycles. Engineers use this to make and improve technology. The same approach is used whether you're making machines, electrical devices, chemicals, or increasingly life. So what happens when we apply this very iterative approach to biosecurity? And I will use that to share with you 4 specific examples of lessons for biosecurity that we've learned in IGEM. You've seen this slide before. I was happy to see Andrew use the same picture. We need to be very clear what we're worried about when developing biosecurity regimes. Synthetic biology often operates at the level of parts, as I've described. These are biological components with specific functions. They're often found at levels below the organism. Yet, when we build our biosecurity regimes, we talk about organisms. That's what we worry about. We have lists of organisms. This list on the slide is just one example. Similar lists sit at the heart of every national control regime I've ever seen. So it doesn't matter whose list I was putting up. This one happens to be harmonized between about 30 countries, but every national list is still based upon this idea of organisms. Forward-thinking regimes sometimes do address parts, but they largely only focus on parts that are only found in the pathogens on the list. But what happens when there's a part that's found in multiple different organisms? I'm going to walk you through what this chart shows you. It is a comparison of the overlap, the similarity between the genomes, the genetic material, of different pox viruses. That's the family of viruses that include smallpox. Looking at the axes, the names in red on the axes are the viruses that are on that export control list I showed you before. Those in black with single letters are other pox viruses from the same family, but that aren't covered by export controls. That cluster of red boxes in the middle shows the comparison between viruses whose genomes are over 95% the same. Looking at that, you can see that there are viruses that aren't controlled that are very, very similar to some of the most feared pathogens on the planet. And the same is true when you look at individual genes. When we did this analysis a couple of years ago, we were able to identify pathogenicity genes. So these are genes directly linked to the things that causes disease in uncontrolled pathogens that were nearly identical to those from smallpox. And one example, one of those genes differed by as few as 11 out of 263 amino acids between the one that was controlled and the one that was not controlled. The sorts of genome or gene editing tools that you will hear about in a few minutes make it relatively straightforward to be able to edit the sequences from the non-control virus to make it into the controlled sequence. It's also become clear that we need to build systems that can deal with parts that are functionally the same as those on the list. Sometimes our control lists do address functions like encoding things like toxins that would pose a significant hazard to human, animal, or plant health, or functions which could endow or enhance pathogenicity. But again, these only apply to the listed pathogens. What happens when a gene has the same function that comes from somewhere else? Early in 2018, an IGEM team contacted the Safety and Security Committee at IGEM and asked about using a part from a pathogen on an export control list. It's this pathogen you can see on the slide. The natural function of the part was to detect those eukaryotic organisms that Andrew was talking about and to secrete specific proteins that were involved in the infection process. But the team wanted to take the part and use it as part of an engineered system to help with secreting the proteins that they wanted to produce. So not secreting anything dangerous, but secreting something useful. The gene is part of what we know to be a pathogenicity island. That's a collection of genes known to be connected to diseases in humans. And they found a supply company on another continent that would give them a safe lab strain of bacteria that had this specific gene added into it. That system could be used at the very lowest levels of containment, meaning that it was as safe to use as the sorts of things that we use inside the IGEM competition. The team wanted to know whether they could use this altered bacteria inside IGEM. So we worked with the team to see whether they would need an export license. We worked with the national officials to try and find that out for them. It became clear during that process that the gene they actually wanted to use, in reality, came from a different but very closely related microbe. This one wasn't covered by export control lists. And our literature reviews revealed that the parts in this non-controlled pathogen and the controlled pathogen were functionally identical. So this biological part came from both pathogens, one on the export control list and one not. So is a part covered if it's functionally the same as the one being controlled for biosecurity purposes? The answer we got was maybe. We also discovered that the process to get a definitive answer to that question was likely to be longer than the entire cycle of the IGEM competition, which meant that in practice, the team couldn't go down that route. So they found a workaround. The team sourced the part from within their own country and therefore sidestepping the need for an export license. Often trying to figure out whether a specific biological part is controlled or dangerous is left to the companies that supply them. For example, there are companies that will print any gene sequence you order. Many of those companies have come together to form a consortium to help them screen their orders and customers. They want to make sure that potentially hazardous gene sequences don't get into the wrong hands. And they've briefed international processes like the Biological Weapons Convention on their efforts in the past. How do we know how good these screening practices actually are? Well, to become a member of this particular consortia, a company has to take a test. They have to be able to identify a number of hazardous orders from a test batch provided by the existing members. But what happens if someone tries to deliberately circumvent that screening system? Several IGEM teams over the years have worked with this consortia and others to perform penetration tests. This example from 2013, this team was working on a project based on something called RNA pseudoknots. You don't really need to know what they are, you just need to know that these potentially encode multiple proteins into a single sequence. The team wondered if they could encode toxins and hide them inside what looked to be benign orders. They worked with two of the member companies from that consortia to find out and established that actually the existing screening technologies were able to identify all the hidden hazardous sequences. A few years later, a team from the same institution attempted an even more sophisticated test. They were working on a cell-free biological system intended to increase the safety and accessibility of synthetic biology for education. Their system included changing the coding used to turn genes into proteins. And that made it virtually impossible to know what a sequence would make in their system. They'd effectively rewritten some of those central rules that Andrew was telling you about. They then worked with 5 of the member companies from that consortia to see if they could hide orders for hazardous sequences. On this occasion, none of the companies were able to identify the hazardous sequences. Much more sophisticated screening software would be needed to address this sort of approach. The team in question shared their insights with the industry and assisted in efforts to close this potential loophole. All of the examples that I've talked about so far involve pathogens and toxins. In fact, all of our biosecurity regimes seem to focus exclusively on pathogens and toxins. But are there other threats out there that we should be thinking about? And our experiences at IGEM suggest there might be. This team from 2018 was working on a project to recycle metals from used electronics. They realized that the microbe they were engineering might also be able to recycle these metals from electronics still in use. They were effectively working on an agent capable of degrading electronics. They spotted the dual use concerns around such a project. They reoriented their project to work only in water, envisaging a use in waste mine water, but also notably reducing its potential for malign misuse. They demonstrated that microbes could be deliberately misused to cause harm other than disease, and they demonstrated that microbes other than pathogens and those that produce toxins can be dual use. Over the last decade, we've learned that it's critical to be very clearly define what we're worried about when developing biosecurity regimes. We need to build systems that can address biological function as well as where things come from. We actually have to test the regimes we build, and we must actively look for ways to improve those systems, for example, by reconsidering core terms and concepts like dual use. Finally, just to say that a lot of our safety and security work is supported through a grant from Open Philanthropy, and we really wouldn't be able to do everything we do if it wasn't for their support. And that's the end of my remarks. Thank you very much.
Thank you so much, Piers, for this excellent presentation that unveils, important aspects of the governance challenges faced by the global generation of young practitioners in IGM and beyond. But I think You showed us specific case studies that help us reframe what we call, dual use elements of concerns, and I think that's extremely interesting to see that this young generation is helping with that work. We will go now a little more in depth on the science and the function of genome editing technologies. The floor is yours.
Perfect. Thank you. So, well, thank you very much. And so over the last few decades, the development of new genome editing techniques has led to significant progress in the field of biotechnology. Of many techniques invented in the past, the CRISPR-Cas9 system is the most versatile of all genome editing techniques because it offers unprecedented ease, accuracy, and efficiency. And the technology of genome editing is all about cutting and pasting DNA, which was already done before. However, the upgrade here is that now we can cut and paste DNA at predefined locations in the genome. Now, the basic molecule that is involved in the cutting of the DNA is called a restriction enzyme. These restriction enzymes work like molecular scissors, which are cutting the DNA 2 times, one on each strand at very specific genomic locations. And the site at which enzyme cuts the DNA is called restriction site. So basically what happens is that these molecules cut the DNA at predefined locations, and these are called restriction enzymes. So when you're cutting the DNA, either it can be a straight cut or it can be a staggered cut. When you have certain enzymes, they cut straight into the DNA, and we call them as blunt ends. And sometimes the cut is not straight, it's staggered, and we call them as sticky ends. In both the cases, it's possible to make a cut and introduce a DNA of your choice. However, in the first case, when there's a blunt end, it's an error-prone joining of the new DNA. But in the staggered one, the sticky end, the joining of the DNA, the gluing of a new DNA fragment is a little more precise than the blunt end cutting. So this cutting of DNA can lead to several possibilities. It can lead to gene disruption, when you have to disrupt an existing gene and check its function, or it can lead to gene correction, or it leads to another gene getting added to the sequence. So the first major challenge in genome editing is really to hit the bullseye. And we need enzymes for that. We need some systems for that. And we know that it is not only the location that we're interested in, but also these enzymes, they may cut the other sites which are not intended. So the first challenge is really to have minimal collateral damage, and that's where the research is happening. So the question is, how do you achieve a targeted integration? So the restriction enzyme recognizes specific DNA sequence, usually between 4 and 8 base pair long. And in an ideal situation, it would be great to have a restriction enzyme that cuts only at one specific spot in the genome and eliminate all the off-target effects. And if you look at the numbers, you realize that in one genome, there can be more than 1,000 cuts. So now the whole idea is to minimize this and make sure that there's a targeted integration. That's why sophisticated techniques like CRISPR-Cas9 come in. So that's, of course, first challenge was to have a targeted cut in the genome. And the second is that once you start feeding the cells with the construct of your choice, most of the cells don't take it up. Only few cells take up this. And therefore, you need millions of cells and thousands of vector molecules to find ultimately the right kind of cell that has a vector of its in its right place. So that's the second challenge. How do you make the cellular uptake more efficient? So the big idea since 90s has been the engineering of DNA nucleases, as you call them, to make the cut at any location of your interest. So this has been observed in the past by using certain techniques called zinc fingers and talons, it was possible to reach out to a certain sequence. However, this has its own limitations, and that's where the CRISPR-Cas9 system comes into picture. So essentially, it is about finding the location. If you zoom into the region where genome edits are supposed to take place, you realize that there is a matchmaking between the guide RNA and the target sequence. So it is like finding the right lock. You have the key, now you need to find where is the lock, and then you unlock that, and the DNA is cut. So the point is that how does it happen? The Cas9 enzyme, it looks for this specific sequence called as PAM, and once the PAM is located, and then the other processes get triggered. It's a little elaborate process, and I'm not describing it in great detail, but just to mention that the location of the protospacer or the PAM sequence is very important, and that is the signature that these enzymes look for. And the moment you see that signature, then the other editing process starts, and therefore it's very specific, and you can introduce these sequences, and it is very much doable to target a sequence of your choice, unlike the other techniques wherein there was a limitation of the off-target effects. So now there have been many applications of this CRISPR-Cas9 technology. People have used it in the fundamental biology, in the biotech space, and also the medicine aspects. People have used this technology to create more variations of the genes, to disrupt the genes, to upregulate or overexpress certain protein products, and thereby generating certain applications. So to make sure that it is more efficient, scientists have designed a variant of Cas9 called Cas5, which is half of the size of the original molecule, and this work has just been published. And the whole intent is that you make more compact Cas9 complexes and deliver them into the cells. Likewise, there was a recent work wherein the group was able to demonstrate, as you can see the right panel, there have been spots on the squid, and using the CRISPR-Cas9 technology, the spots were removed. So there was a very powerful application of this technique. And also recently, this was posted recently by Dr. Francis Collins, and this was about editing the sickle cell related cellular system. The sickle cell disease is caused by a specific mutation in the gene that codes for beta chain of hemoglobin. And therefore, the researchers try to correct the sickle cell, as you see here on the left panel, and then you see a corrected version. So this is a very powerful demonstration of an application of the genome editing technique. And I believe the human clinical trials are on the way. So like that, now people are trying to use bacterial toxins for DNA surgery. And there are areas where CRISPR-Cas9 system is unable to reach. And therefore, certain bacterial toxins have been found which can reach mitochondria and do the editing. So to make sure that the governance aspect is well taken care of, the expert advisory committee developing global standards for governance and oversight of human genome editing has been created, and there's a lot of work going on in the background. I think we'll talk about it very soon. So over there in our university, Professor Singh has been able to use CRISPR-Cas9 to reduce the transmission efficiency of malarial parasites in mosquitoes very effectively. So this is a good use of the CRISPR-Cas9 technology. In our lab, we have made proteins from sequences that were not destined for making proteins, the not coding sequence. And so we have demonstrated that sequences that were not designed for, and this has led to several applications. So we recently made peptides from the tRNA called tRNA encoded peptides, and it was amazing to see that they are very powerful. This set of peptides that we made, they're very powerful anti-leishmania peptides, and this gives rise to a new area wherein you're trying to make novel molecules from sequences that nature did not use for making proteins. So finally, we have found some Cas-like molecules from the sequences between the genes, and we're also looking at anti-Cas proteins. Well, thank you very much for your kind attention. I'll be happy to take questions.
Thanks, Professor Dar, for this excellent talk, which is illuminating, a lot of the functioning of potential applications, but also giving us, concrete example of what's possible not only in the medical realm, but also the example about suppressing the transmission of malaria parasites in mosquitoes. It's actually a very interesting application in the non-human genomics. So we'll now move to the second phase of our panel to explore with more depth some of the technical challenges that scientists, private sector actors, and policymakers need to overcome to modernize and improve our capacity to control dual-use biotechnologies and ensure international security. And I would like to recognize a question that came from a researcher at GNU, Akansha, and I'm using this question as a general frame because I think it gives us a good sense of all the actors that need to be involved in arms control. So the question is about how countries, governments, militaries, and other forms of leadership should prepare against the intentional biological risks. So what form of national biodefense strategies should we be thinking about? So just have those types of actors in your mind, and I'm going to go through a few more in-depth questions. So let me first start with a question to both of you. What feasible threats are posed by genome editing when it comes to conceptualizing, designing, and producing potential biological weapons? So how does the convergence of functional genomics and gain-of-function research, for instance, influence potential misuse? But also, specifically for Professor Dar, what are the technological, delivery, and knowledge-based limits faced by practitioners that may prevent a weaponization scenario? So maybe we start with you, Professor, and then I'm eager to hear from peers too.
All right, great. Yeah, well, there are technical challenges to the CRISPR-Cas9 system. It's not a perfect system yet. There are, of course, possibilities. It's a dual-use technology, and it can be used in certain ways. However, in my view, the threat level is not extremely high because it is a fairly complex system. When you're playing with the organisms, this is billions of years of evolution, and so you cannot beat that intelligence just with a couple of experiments. So therefore, it needs a lot of work to actually understand how the whole organism is working and do something with that organism. So it is possible, of course, to go in that area of creating some nasty molecules. However, there are more technologies that can do. There are cheaper methods of making some nasty biological weapons. CRISPR-Cas9 still needs a lot of attention because it is not a perfect technique yet. There are off-target effects, and so it needs a lot of work.
Piers, do you want to give your insights on this too?
Sure. I actually think the short video that was played during the break before this session started was exactly right. And if I remember correctly, that called out two specific elements. The first one is that you can use it to resurrect or recreate things that we already know are dangerous, to print viruses or to some extent bacteria, as Andrew was talking about earlier. And that can circumvent some of our control regimes. I talked a little bit about the challenges of being able to screen gene synthesis orders and to control pathogens in a day when you can print them and reboot them from scratch. And so, for example, smallpox is only meant to exist in 2 laboratories in the world, one in the US, one in Russia. but the paper was published by some Canadian researchers last year, demonstrating that you could synthesize a very, very close relative of smallpox. So definitively proving that we can synthesize any virus that we wanted to. And then the second issue that the short video picked up was the potential to use these technologies to make things that we haven't seen before. The sorts of engineering-based approaches to create organisms with specific function. We are getting better at being able to do that for many beneficial purposes, for using biology to make things. The bioeconomy, as Andrew mentioned, is somewhere between 2 and 3% of GDP in many countries, and many of our countries around the world. And this really isn't just developed countries. This includes a significant number of low and middle income countries are investing incredibly heavily in their bioeconomies. So our governments writ large believe we can make things with specific purposes out of biology, and one of those purposes could be to cause harm. So I think those are the two things that I would say at this point. Thanks.
Yes, I will pick up on what you just, you kind of hinted at some of the limits in our regulatory framework. So we increasingly measure the shortcomings of control lists based on a taxonomy of specific non-bioagents, and increasingly a more nuanced paradigm might be to control genes based on their predicted function. So for instance, the ability to screen sequences of concern, including genes responsible for the pathogenesis of viral threats, bacterial threats, and toxins. Yet function-based restrictions still raise important questions about what functions should be regulated and how confident function predictions need to be before they justify the control of a sequence. So what are the limits in applying functional genomics to screening of biological threats, and how could genome editing help overcome these limits? It's for both of you, Piers and Professor Dar.
Okay. Piers, you want to go first, or?
Happy to. I've got some thoughts on some of these things. I think taking the questions in reverse order, I think one of the most famous biological warfare agents has to be the causative organism for anthrax, Bacillus anthracis. What is it about that bacteria that puts that pathogen on all of these control lists? Why was that the one chosen to put into the letters? Why was that the one weaponized by the UK during the Second World War? In part, it comes down to two biological functions. Firstly, it makes a nasty toxin that makes people ill. And secondly, it's incredibly stable. Its ability to form spores makes it hardy, and you can put it out in the world and it won't die like many microbes. Each of those biological functions, we know the genes are responsible. We know that those genes are found on plasmids inside those bacteria. And yet, those control lists that I was talking about refer to this thing as Bacillus anthracis. Now, the interesting thing is those two plasmids also exist in really closely related species. So there are closely related species that has both of those two biological properties. And yet, because it's not called Bacillus anthracis, isn't included in our control lists. The noticeable exception is the US updated the select agent list a few years ago to include, and I won't bore you with the scientific name, closely related bacteria that have these properties, the same properties as anthrax, Bacillus anthracis. So in a way, they're already putting the biological functions into the lists. And so I think we've demonstrated we can do it. In that case in point, we need to think very much more about what are the, I think you got the question right, what are the functions that worry about us? When some of that's obvious, that being environmentally stable, it may be being able to be inhaled into the lungs, it may be producing nasty toxins, or as three examples. So that's that part. And then the second part, I answer the other thing you were asking about, how are we moving towards being able to predict function or predict whether we need to worry about apart from its sequence. There are a number of ongoing research programs to do exactly that. Two that spring to mind. They're both run by the Intelligence Advanced Research Projects Agency in the US. Projects called Felix and FungiCat. Felix is designed to detect signatures of non-natural origin, that to be able to spot whether something has been engineered or as opposed to evolved naturally. And then the second one is to be able to identify potentially hazardous functions from pathogens, with the idea of creating algorithms that were able to predict a hazardous function from a completely unknown sequence. So something that we've never seen before, that where you can't go back and say, yes, this comes from a known pathogen, therefore it might be dangerous. Or this looks very similar to a toxin made by a different pathogen, therefore it may be dangerous. But something that is completely novel, but still potentially hazardous. And both of those programs have had successes. They have resulted in papers being published. That information is in the public domain. So I would argue we are moving slowly but surely in that direction. And 10 years ago, a big Academy of Sciences study in this area felt that these capabilities were so far over the horizon they weren't even worth talking about. So in less than 10 years, we've moved from so far over that horizon we can't imagine moving to function-based systems, to algorithms that are proving useful in detecting non-natural origins and potentially hazardous functions. So I think we're getting there.
Thank you so much, Piers. I will add, we just got a very interesting question from the audience that's related to what we're talking about. So I will add it to our discussion, and maybe we can tackle it in the answers. So I'm being asked to see with you if there are strategies that would allow for the control and restriction of subgenomic portions from non-regulated pathogens that also overlap with regulated pathogens. So this problem underlies the synthesis of horsepox from synthetic nucleotides. Are there any easily implemented methods that Piers or Professor Dare that you see that could have prevented the horsepox situation?
Great. I think it's time to work on these aspects which have escaped the attention, largely because the perception of threat was something different. Now, a couple of years back, a new area has been started, and that is on the anti-CRISPR proteins, ACRs as we call them. And DARPA has a big program on that. So you create an inventory of molecules that can prevent the CRISPR proteins from binding the DNA. That's one set of molecules. And the other set is to prevent the CRISPR complex from cutting the DNA. So that's another area where people are focusing on. So anti-CRISPR molecules is a hot area currently. And in that sense of genome editing, I see a lot of activity in that area in the near future.
Mm-hmm. Piers, do you want to comment on this second question I mentioned that's kind of related to functional genomics? Do you want me to repeat it?
If you don't mind, that would be wonderful. Yep.
So what would be strategies that could allow for the control and restriction of subgenomic portions from non-regulated pathogens that also overlap with regulated pathogens? So you mentioned this question of the homologs in your presentation. And this problem underlies the synthesis of horsepox from synthetic nucleotides. Are there any easy implemented methods that would have prevented the horsepox scenario?
It's very interesting. But it's a really, really interesting case study, because my understanding in many countries, it would not have been legal to do some of that work. Because of some of the genes used to make horsepox are so similar to the genes that are found in smallpox. It is generally not permitted to work with genes from smallpox. And because it's in both, it makes it very difficult. It may not technically be covered by an export control rule at that point in time, but the rules around smallpox are unique. That problem, because there is an international regime in place for that virus. For any other virus, this problem is blown up even more in case. I think the important thing in these scenarios is the companies involved are already screening these orders. They have to do that from a compliance point of view to make sure that they're not sending people things that they're not legally allowed to have, or that they have the appropriate paperwork to have them. Revising, updating, and improving the databases those companies use to screen against is a very practical step. There are ongoing efforts right now, I'm involved with that, to create a harmonized screening database and set of tools that could be used in all countries for free, ideally, to allow all companies to do that sort of screening and all institutions that want to screen to be able to do that sort of screening. So the question is as simple as, if we identify things we don't want to make readily accessible, we can add them to a screening database, and making that as freely available as possible will help us do that sort of work. Certainly, I spoke a little bit about the fact that IGEM screens all of its parts, so we have a system to do that. We're very lucky that our commercial partners have provided that for us in lieu of sponsorship for IGEM. The costs involved, if we had to pay a commercial rate for that, would be prohibitive for IGEM itself. We take up a good number of hours directly of a very senior vice president from a very influential biotech company whose time is incredibly expensive. So I think those tools are what I would highlight the most. I know it's not an easy answer or a concrete answer, but it's also something that we haven't really looked at having on a multilateral level, and that may be interesting to do.
Thank you so much, Piers. We had actually received a question from Pamela Moraga, who was, kind of notifying with us the shortcoming of the existing regulatory framework and asking what states can do, can take as alternative strategies to enforce the non-proliferation regimes. And I think you brought up, the role of the private sector, and especially the updated tools, processes, and mechanisms in preventing illicit gene synthesis and other advances. So I think that's really that's convenient, and that really answers also this question. I had two last questions for each of you, but I think we'll have to do it very quickly. I wanted to balance a little bit and talk also about the opportunities that genome editing provides us with. So I wanted to ask Professor Dar about opportunities for mitigation. Is there a significant role for genome editing technologies in improving medical and biological countermeasures to protect against and mitigate against biological threats? And then, Piers, for you, I would have a question about, you mentioned the threats identification algorithm that we can use for better understanding, better predicting functions of combination of gene sequences. How sophisticated is our knowledge and our approach to this convergence of AI and genome editing to be able to use such algorithms? You also mentioned in your presentation the possibility of encrypting some sequences by gene recoding. So we're facing new challenges at the intersection of cyber threats and biological threats. If each of you could tackle that last question, and then I'm going to wrap up because we'll be over time. So for you, Professor Dar, it's about the medical and biological countermeasures. What role for genome editing in that mitigation strategy, or that on that aspect?
Great. I think it's very important that it is, first of all, we need to understand who are the players involved in this in order to mitigate or in order to control or manage it more effectively. So the first and foremost requirement is the information. Who is the user and for what purpose they're using? Because what is going to happen in the next few years is that these tools are going to become more sophisticated, not only in the clinical application domain and agriculture, but also multiple edits are going to be performed at the same time. So which means that the power of CRISPR-Cas9 is going to improve over time. So it is all the more important that we should first of all know the players who are involved in this, And at the regulatory aspects, at the funding level, one is to see, what kind of projects are being funded, because still it's at a nascent stage, and it is largely a self-regulated community. So I would say, along with CRISPR-Cas9, what needs to be regulated is the long DNA synthesis, because it might take some time to perform the precise genome edits, but long DNA synthesis is so fast, it's so quick. And imagine if you have a DNA printer in the lab, you can synthesize the sequence of your choice. So it's very important to get an idea of what kind of work is going on, and then make sure that these are somewhere followed, and in terms of the applications and also in terms of the funding avenues and so on.
Thank you so much, Piers. I don't know if you remember the question. It's a little bit more about that convergence with threat identification algorithm and our notion, our knowledge of functional genomics. What do you see, coming up in the next 5 years that could help us, better modernize our non-proliferation regime?
Just a small question, then.
Yes.
So those algorithms are at very early stages, right? They have succeeded in the proof of principle steps. They are a long way off from being able to accurately predict hazardous function from sequence in a reliable manner. But I would just re-stress, in 10 years, we've gone from impossible to having them. So I would imagine in the next 10 years, we're going to go from very early stage to something a lot more sophisticated. The second thing I would say, sort of coming to this overlap, in particular with machine learning, there are bits of biology that seem very suitable for artificial intelligence and machine learning approaches. For example, around proteins. So we spoke a little bit earlier about how genes are turned into proteins. The physical shape of proteins is incredibly important for their function. And machine learning has proven particularly good at being able to look at proteins we know about and learn lessons to predict the shape of proteins for a specific function, and to make proteins that have never existed before, naturally, that do what we want them to do. And so if you think back to what Andrew was talking about this morning, about that central tenets of biology, if the proteins are at the end, the step then is, can we understand how the genetic material is translated into proteins in such a way that we can use machine learning to go from the sequence right the way through to the biological function. And that is not something that's necessarily here at this point in time, but it may be that those sorts of approaches, when you combine them with genome editing tools that let you copy, edit, remove, replace, add in genetic material, would give you a powerful suite to do the sorts of things with biology that Andrew was promising us we're going to see at the start of this meeting.
Thank you so much. That's really, really interesting insights, exactly what I was looking for. So I'm going to close this panel. We're a little bit over time. So I'm going to close with a few takeaway messages. First, the notion that modern biotechnology, including genome editing, provide an increasing and maybe later exceptional potential for improving our knowledge of functional genomics and proteomics, as Piers just mentioned. And applying functional genomics approaches to biological threats might be a strategy to modernize the arms control toolkit. Yet many important questions remain about what functions should be regulated, what algorithmic and bioinformatic tools can help us achieve more precise and comprehensive screening. Increasingly, such questions will involve the convergence of computer and life sciences, as well as cyber biosecurity challenges. Second takeaway is that practitioners using genome editing technologies form a truly global community, which includes the next generation of researchers, private sector actors, and policymakers. And so there is an imperative need for more foresight, red teaming, and collaboration, as we saw very well in the IGEM examples, at the intersection of these sectors to better manage access to knowledge and technologies. And relying on the design, build, test, and learn cycle might help structure efforts to reinforce biosecurity, as shown by Piers. Finally, at a time when public concerns over biosafety and biosecurity are crystallizing, a special policy translation and education efforts are needed. And I think our panelists and UNIDIR show excellent leadership on that front. So many thanks to all of you. Giacomo, I give you the floor back. Thank you so much for all your insights.
Thank you, Eleanor, and thank you, Piers, and thank you, Pawan, for a very engaging panel. It was very fascinating to hear your presentations and your discussion. The next panel will explore how the evolving research and development landscape, both in terms of new technologies and new actors, is really democratizing and accelerating the cycle that has been discussed extensively already, the design, build, and test cycle.
Hello, everyone, and welcome to our next session on cloud labs and DIY bio. I'm Jamie Yosef, and I'm a senior fellow for global biological policy and programs at NTI. I'm a biophysicist by training, and my work is focused on biosecurity and pandemic preparedness. We have a great discussion lined up for the next hour, but at the top, I'd like to just remind everyone that you can submit your questions in the chat function. As we move through this discussion, you do not need to wait to the Q&A at the end. So over the next hour, our panelists will describe the work of DIY biolabs and the cloud lab industry, and how these very different types of laboratories operate. These are both exciting communities where a great deal of innovation is taking place, which is tremendously valuable for advancing public health and economic development goals. We will also discuss some of the great work that each of these communities has done to proactively address biological risks associated with this work. We'll share some thoughts about how these risks may evolve over time. And I think at this point, and as we've been discussing throughout the morning, it's worth noting that this discussion fits within the broader context of rapid advances in technology and the emerging biological risks associated with this work. Life science and biotechnology advances are critical for reducing pandemic threats, addressing health challenges, and supporting economic development. But they also pose unique risks and have the potential to undermine global progress towards these very same goals. Rapid advances in the fields of bioscience and biotechnology have made it difficult for governments to keep pace, which has meant that academic research institutions, biotechnology industry, community laboratories, funders, and others will need to continue advancing innovative solutions to safeguard bioscience and biotechnology. Engagement by the international community will also be vital in accelerating progress in biosafety and biosecurity. And so I'm so thrilled that UNIDIR is convening these discussions today. Within the UN system, the World Health Organization, the Biological Weapons Convention, the UN Office for Disarmament Affairs, and others all have a critical role to play. But there is currently no dedicated international entity that is fully focused on this important work. Now, driving continued progress on biosafety and biosecurity is essential, not only to continue to safeguard DIY labs and cloud labs and all the benefits that come with that innovation, but also to protect the broader bioscience research and development enterprise. And fundamentally, the ultimate goal of this important work is to guard against exploitation of the legitimate global bioscience enterprise by malicious actors who may seek to cause harm through biological attacks or other means, and also to reduce the risk of an accidental laboratory release event with potentially catastrophic consequences. While naturally emerging novel pathogens can cause severe damage, and as we are currently experiencing with SARS-CoV-2, engineered or synthesized pathogens may have the potential to pose even greater risks to human health and economic stability. We are very lucky today to have two excellent panelists who can help guide our discussion about these important issues. Todd Kukin is a senior research scholar at the Genetic Engineering and Society Center at North Carolina State University in the United States. Todd has been an active member of the synthetic biology of the DIY bio laboratory community for years, and he's played an important leadership role in ensuring that the work of this community continues to be safe, secure, and responsible. We'll also be joined today by Peter Lee. He is the Director of Science and Technology at Stratios, which is a cloud lab company based in California. Peter is going to share his thoughts about the innovative work at Stratios to advance automated approaches which can improve science and open the door to investigation at scales and speeds that have not been possible previously. There's a lot of additional detail in the list of bios about our great speakers, and so please feel free to review those at your leisure. But in the interest of time, we'll turn it over to our first speaker. Todd, over to you.
Thanks so much, Jamie. And I want to thank everyone for joining us. So good morning and good afternoon. I'm going to try to kind of give you an overview of the growing global community bio world, kind of hopefully from its origins, and talk about some of the work we've been doing to address some of the safety and security concerns. I want to start, though, with a shared vision statement that was developed at the second Global Community Bio Summit that's held at the MIT Media Lab, and which started a few years ago. And this statement was put together by the over 200 or so individuals that came from multiple community labs and groups from across the globe. And basically what they're stating out or laying out what it is that they're trying to do and their vision. And what they're doing is fundamentally transforming the life sciences and democratizing biotechnology to both inspire creativity and improve lives by organizing life science change makers and bioenthusiasts to try to build an inclusive global network, cultivate an accessible commons of knowledge and resources, launch community labs and projects, and enable local educators. So where did this sort of begin? So some of the origin story, so to speak, at least in the United States, at least the one that we have with a picture, was back in 2008 in a pub outside of Cambridge, Massachusetts, which also happens to be a hub of synthetic biology, which we heard Andrew Hessel eloquently lay out earlier this morning. And it's interesting, these groups of bioenthusiasts met to really start talking about how more people can start to access these tools and information of biology, and particularly biotechnology. And if you really dive into those pictures, you will see individuals like Tom Knight, who some can argue is sort of one of the sort of founders of at least the term synthetic biology, as well as some of the early sort of developers of the International Genetically Engineered Machines competition. And what was interesting was that similar events were occurring across the globe, where individuals were meeting in online discussion groups, all talking about how they were able to sort of begin to work with biology and utilize some of the tools as the costs were really dropping rapidly. So what was happening was the access to the tools of biotechnology were becoming, in a sense, accessible. And people were then asking the question, well, why not me? Why can't I also embrace and learn and innovate with these technologies? Shortly thereafter, in 2010, we had the first community biotech lab, which opened in the United States. And there's an argument, of course, which one opened first, but let's say there were two, one on the East Coast and one on the West Coast, that basically opened at the end of 2010. Since that time, we've seen a really rapid growth in the number of these labs that have occurred across the globe. And you sort of see this inflection point, really, I would say, around 2013, where you had in the tens, and now we're over sort of about 50 or so, we believe. Some will argue it's upwards to 100. Don't focus too much on the exact numbers in these charts because these numbers are extremely fluid. But what also has been happening inside these community lab spaces is that startup companies have also begun to spin out of these spaces as well. And those numbers, again, don't focus too much on the exact number, but more look at sort of these trend lines that are happening. And what we think is happening is as more people become educated and have access to these tools, particularly in these community spaces, it's opening the doorway to different innovation pipelines, basically on-ramps to that innovation world, where people who previously might not have had access to that are now being able to actually participate in this world of biotechnology. This is a map that comes from the sphere.diybio.org site, which is one of the sort of hubs of where the community comes together. to list their projects, to list where their labs are, what they're working on, what types of startup companies might be sort of starting as well. And it's really sort of a global meeting place for the community to get together. And I show this map really just to show that this really is spreading across the globe. And what's interesting, if you sort of overlay this map onto the IGEM team maps, there's a lot of sort of similarities there of where this work is happening. And what's exciting is that it's not just happening in the typical sort of tech centers in the global north, that it really is beginning to spread across the globe and has become a global community. So what do these spaces look like? There's no one that looks the same. So I'm going to bring you through a couple of slides of spaces that we've been visiting over the last couple of years with a grant from the Open Philanthropy Foundation to really explore where the current state of the community bio world actually is. So this is a picture from Melbourne, Australia. That's actually a shipping container, which has been sort of converted into a fully working biotech lab. And if you look at that sort of square yellow sticker that's on the left sort of side of your screen there, I'll zoom in. This is actually a certification certificate that comes from the Office of the Gene Regulator in Australia that has certified this lab to actually be able to do genetic engineering. And we'll probably get into this in our discussion period later, but different countries have different sort of rules around who is allowed to actually sort of operate a biotech lab. And this is one of the questions that the community struggles with as well, is this type of certification or approval process that may need to be instigated. This is a look inside that lab. These are some pictures from an actual home garage lab. And as you can see, very similar types of equipment that you would find in any college lab, although some with sort of some hacks. So that is sort of a pressure cooker there that you would find in your home that can double as an autoclave. This is a picture from the lab in Baltimore, Maryland in the United States. Again, fully functioning, that basically you would find everything in here that you could find at a typical biotech lab on any college campus. This is a designer who has been helping to actually design these shipping container labs and incorporating the idea of design in terms of the flow of how these labs actually look and operate. You also can find them inside libraries and also behind churches. This is a photo from Georgia where there's a community lab inside a larger makerspace, which you may be familiar with, where they have carved out a component inside that makerspace for a biology lab as well. One of the first things the community did and that we helped them with all the way back in 2011 was to develop these codes of conduct. The community from the very beginning has recognized sort of their responsibility with the awesome power that comes with working with biology. And so they came together both in Europe and in the US, which were the two main hubs at the time, to try to lay out how they were going to operate both safely and responsibly. That has developed over the years, particularly with this project we've been having that was funded by Open Philanthropy, where we've designed a biosafety bootcamp course. We partnered with the International Biosafety Officers Association to develop a sort of train the trainers course to be able to train labs and lab heads how to actually run a lab safely and responsibly. So these folks came from 15 different labs, again, from Europe, Australia, Africa, and the US. And they went through a 3-day course learning about all the various different types of safety and security measures that you would want to put in place inside of a laboratory. So you might not be able to see this, but they went everything from risk assessment, hazard identification, chemical storage and disposal, what you can transport, what can't you transport. And the idea of this is that these folks then go back to their own communities and can train their members inside those labs to adopt these practices. So all of that is to say that the community has taken this responsibility around safety and security very seriously since the very beginning. They've recognized sort of both the innovation potential and the access points in terms of what it means when you can actually educate people around these tools and techniques for people who had never had access to or might never have access to this type of information. And they really are serving as sort of these educational hubs, almost science education hubs in communities where if you didn't have the sort of finances or privilege to go to a university, you may never have actually sort of interacted with this science before, and now you can. So the other question, though, is, what about in the midst of a global pandemic? So what we've seen from the community, particularly on the Joggle site, which is a new platform called Just One Giant Lab, which is geared towards getting these communities and community labs to work collectively on projects, again, spread across the globe. And they were very quickly able to begin an open COVID-19 initiative, where groups across the globe are working on everything from personal protective equipment, diagnostic tests, and also vaccines. And what's interesting about this is, again, from the beginning, which is some of, I think, credit to the work we've been doing over the last 10 years, is they instituted a safety and security board from the beginning. But interestingly, all of this is being done sort of virtually, and actually we're on Slack. And so we received a grant from the US National Science Foundation to actually study this question of how do you actually do ethical research around things like COVID-19 vaccines in an open, virtual, disconnected community of people working in separate locations, but working towards the same goal. So we're trying to study how these issues come up in real time and how they can become addressed. But you may also have heard about folks that have also developing their own sort of vaccines and injecting themselves with it. Like any community, not everyone is going to sort of abide by or agree to all of those same terms. And so, however, I think what's important is that for the larger sort of community sort of is trying to address these issues in a clear and responsible manner. Interestingly, about this particular project, which is leading by Josiah Zahner, who has done these types of sort of stunts before, is that they wanted to make it clear that this is about an educational project. They're actually not trying to sort of distribute a vaccine, that they're actually using this as a way to teach people how you can actually sort of learn the science behind developing vaccines. But at the same time, we have sort of this blurring of the lines as well, where we have another project by the RADVAC organization, who are, what you would call traditionally trained geneticists and scientists coming out of Harvard Medical School, MIT, who have started these projects outside of those institutions, working in their own garage labs, and then distributing sort of the recipe and know-how on how to actually create these vaccines. And yes, they have been taking them themselves and have been encouraging others to take these to, in essence, run their own sort of pseudo types of trials to figure out how these vaccines may or may not work. And I thought the interesting quote from one of the heads of this was that they're going for engaged consent, which goes beyond informed consent. So I think this combination of sort of this growing set of community biotech labs, sort of garage home labs, and then this blurring of the lines of so-called sort of traditionally trained scientists who are operating sort of outside of institutions raises questions about sort of how do you actually sort of manage sort of the safety and security risks in a community that isn't necessarily captured by any sort of top-down governance system. And what we've been trying to do over the years is do a bottom-up type of governance system where these types of norms, ethics, and philosophies are embedded inside the community. So with that, I will end. I just want to end with this photo here because it's not just sort of typical scientists, even garage scientists that are using these tools. The art and design community are also embracing these tools. And so I encourage you to go to these links here and look at this catalog of an art exhibit that we put together, which also shows how artists are beginning to utilize biology, synthetic biology, not just as a reflection on the science, but also in the actual artwork itself. And so when we're talking about safety, security, and governance, we really need to look at the entire spectrum of who now has access to these tools. So with that, I will pass it back over to you, Jamie. Thank you very much.
Thank you, Todd. Thank you for an excellent presentation and for a lot of thought-provoking comments, which we will certainly revisit during the discussion session. We will now turn the floor over to Peter Lee, who's going to share his experience working in the cloud lab industry. Peter, over to you.
Great. Yeah, thank you all, and thank you all for the time. I'm really grateful to be able to join this conversation and really talk about how innovative technologies are out there and discuss their security implications. And as we'll, I think, discuss through this hour, these conversations are really important to be having basically early and often, and we really enjoy participating in that process. So today I'll be telling you about cloud labs from the perspective of Stratios, which from what we can tell started the first bioscience cloud lab over 8 years ago in 2012. And since then, we've been working to create easier interfaces so that scientists can access complex life science automation and accelerate their scientific discovery. In terms of my journey, my research has always been focused on incorporating science with software so that we can augment scientific discovery. And from my perspective as a field of biology, as we continue to amass large amounts of data, we really need a way to incorporate all that data and unify it. And for me, it's been a lot about focusing on standards as well as on platforms that can standardize experimentation, allow for repeatability, and also be a resource so that scientists can leverage that for scientific exploration. And that's what brought me to work on cloud labs. So at the heart of cloud labs, I think just in general, it's working to advance life science research and discovery by integrating automation equipment like plate readers and thermocyclers and liquid handling capabilities so that it can be, put together in basically a laboratory operating system. And that operating system basically consists of device drivers that are communicating with these physical devices, scheduling and planning capabilities, a formalized protocol language and protocol creation tools, and overall the ability to collect and coordinate data and metadata and surface a programmatic interface so that in the end, we can mate that to a cloud interface that scientists can use to access replayable, repeatable, and reproducible protocols that can really be used anywhere in the world. So some of the inherent values that arise from cloud labs are that they allow for this interconnected collaboration. And when I'm talking about that, I'm referring to both collaboration among scientists as well as collaboration among facilities. So among scientists can actually use the cloud lab to work with their colleagues across the globe and basically initiate that from the comfort of their own homes, which I think a lot of us have gotten very familiar with over the past couple of months. When we're able to physically have conferences, cloud labs will also allow scientists to collaboratively start thinking of experiments from conferences, from talks, and allowing them to gather and really work together to get experiments launched and see results and iterate on that. From the facilities perspective itself, cloud labs also allow you to really basically build once and deploy anywhere. So you can create a network of connected automation labs and develop your protocol in one location and beam it out to all other locations. One of the really nice things about this is you could see that protocols can be improved and iterated on over time and really just propagate through the network. And one thing that we're acutely aware of is that this is happening manually in the world today in terms of our response to COVID. But I think the vision is that this could be happening at the speed of basically software updates and software upgrades. So going back to one of my original aims of unifying biological data, cloud labs are also really great because they're inherently AI compatible and they're amenable for large-scale modeling, model building. Basically, because cloud labs are linking and strongly linking data to formalized protocols that creates that data to the additional metadata that's been collected along the way, we're basically inherently following the fair, findable, accessible, interoperable, and reproducible data principles. And these are the principles and the standards that really allow us to provide clean data that can be useful for machine learning and computational model building, and really allow us to think about how you can have a completely computationally guided iterative workflow for discovery. It's also kind of interesting that as we're really basically starting to build better models already through the protocols themselves. You can kind of think about that as as we kind of iteratively optimize these formal protocols and we start understanding the constraints of those protocols, we really are basically modeling the underlying biology itself. And so this is kind of an interesting way of linking the macroscopic, the protocol layer, what's happening at that level, to the microscopic, to the physical properties of the biomolecules themselves. One of the other things that I think is of value is that as we start to miniaturize and parallelize and really accelerate experiments, our goal is for science to be able to undergo sort of a similar transition from computers that happened in the 1950s, where computers went from being people who computationally performed calculations to silicon chips, to large server farms and mainframes. I think importantly, this is kind of hopefully the transition from what's happening day to day in lab to work cells themselves. This isn't just, I think one of the nice things is it allows for repeating what we've done in a faster, less error-prone way. But I think the real benefit of that is being able to go above and beyond to see what emerges, because we can do things differently now, and we can really think about science differently. And so this is like in the same trend that computation went towards AI itself and virtual reality, complex modeling, the internet. We're really hoping that the advent of cloud labs is also driving this acceleration of science and brings about a similar transition where scientists can really start thinking differently about how they use these tools. So what's Stratius doing in the space itself? Over the last 8 years, we've basically created a laboratory operating system that's integrating automation with an open source formalized protocol language that's called Auto Protocol. And we license that operating system to facilities, and we also deploy it on our own facilities. Currently our systems are covering about a third of an acre, or about 1,350 square meters of lab automation. And this is spread across 2 facilities in the San Francisco Bay Area, as well as in San Diego, we're using the cloud interface to give scientists unified access through to both of those facilities. We've currently been focused on bioassays and small molecule discovery and synthetic biology, and we're expanding currently to automated chemical synthesis so that we can really complete this loop between small molecule production and their effects on biology, studying their effects on biology and repeating that. Really, the whole point of this is that we can provide the tooling and instruments and workflows that may not be readily accessible, or may require large capital expenses, or may have maintenance and quality control debts that then can be distributed to scientists who can leverage that capacity and without having to worry about all the cost and expense of that, so that they can really perform their experiments. And to that end, we're working with biotech startups, larger biotech companies, healthcare systems, pharma companies, and government agencies. So let me show you a little demo of sort of what it looks like to be a user on our system. And when you log into the Stratius web application, you'll be greeted by this projects view. And this is basically, each project is a folder that you can use to organize experiments. And you can see in the projects view that there's the number of runs in queue, the number of protocols in queue, and the number of runs that have been completed for each. In this view, you can also switch over to the inventory management, and in that view, you can start looking at the different containers that you have within the system. So this is kind of virtually managing that inventory. You can click in on that container and get additional information about what's going on with that aliquot. So you can see what other runs, the runs that it's been linked to already, and then you can also click in, get additional information, and perform like shipping that container or destroying that container as needed. So in terms of shipping that container, you can check on Stratios and see what's being, you will need to send to Stratios, as well as what's needing to be returned. And then this packages view is kind of interesting. This is basically our protocol packages. So this is showing you that we're really treating it like code. And so for any given protocol, you can see the number of different releases, the versions of that release, and you can go down and see what all is linked, what other protocols are linked, what other runs, et cetera, are linked to the protocol. So going back to the package projects view, we'll click in on a project and we'll start a run. So for this demo, I'll switch to a new organization that has basically a protocol that that has a simple interface, but reveals a lot of complexity. So we'll click to browse protocols and we'll search for the thermal shift assay and click on that. And that'll bring us to an overview of that assay. And so we can see here that this protocol is going to assess the thermal stability of our protein of interest after we incubate it with a series of experimental compounds, a series of small molecules. And as part of the summary, you'll see that we need to provide compounds, and we also need to provide the protein that we're going to test with. And as output, the data generation portion, we will get this thermocycler data back that will tell us the thermal stability of our protein. So we'll click on the getting started, and we'll see two fields. And so the first input is for the compounds, and the protocol says that we can select up to 21 compounds. We'll click there and choose this compound plate. And we'll just choose 8 for today, but you can kind of choose any up to the 21. Submit that in. And then for the protein, we'll just choose the first tube of protein over here, our thermal shift protein for the demo. And then we can validate that. And so in the back end, this is launching the protocol and checking all of these assumptions and logic and making sure that the inputs confirm. And so once that's validated, you'll get a summary view of the experiment. And you can click through that and scroll through that and see all of the different, basically your work list of tasks that are going to be done in this summary view. You can also click in on individual instructions like this acoustic transfer instruction, which is transferring 200 nanoliters of each of our 8 compounds to the experimental place for that testing portion. And basically at the end of this, if we're happy with this, we can hit the submit button, and that'll get the run actually queued up on our backend systems. So here we can view that run and basically follow along in real time. And right now these instructions are all in white, and as they get processed, they'll turn green. So that kind of is a visual indicator of how that goes. And you can see, because it's code, that you can also clone runs. You can also see, again, the parameters that were used to start that run, etc. So this is kind of all the nice attributes of runs being code. So we'll shift over to basically what's happening on the back end, and I'll show you like a sped up view of what's happening on the work cells themselves in a second. So this you can see the work cell is taking in plates. Basically, in the back end, there's a scheduling process that's telling all of the devices where to go, to open and close their doors, to execute different instructions, to rotate through. So that's just basically a very broad view of what's happening on the work cell side. And then here, this is at the end of that, you get your results view, and you can start seeing basically this is qPCR results, that we've got a standard curve in terms of DNA, and we're measuring the amount of DNA that is through qPCR. And this is kind of similar to how a COVID assay would run. So you can see on the plate map below that every column 1 through 6 is more dilute concentrations of DNA. And so you can see on top sort of a qualitative assessment that each column is, I guess, distributed, has low standard deviation between the columns, and you can see that each column is in a different grouping within that qPCR test. So, of course, this is all programmatic, and you can download this data through the web interface and basically use that for further analysis. And I've shown you basically a graphical user interface that we use here, but all of this is using standard APIs on the background. So you'd also be able to interact with it programmatically, and we have customers that do that too. So really, there's a lot about cloud labs, and on the whole, they're leading and moving and accelerating the space. And in that space, we really want to be conscientious and careful developers, and so we're interacting with the biosecurity community basically frequently, and thinking about best practices and overall coordination. For Stratios itself, we're taking a multi-layered approach to security. So we're ensuring that we're working with trusted partners within the scientific community. We currently work closely with those partners so that they can take their protocols and we basically translate them so they work with our operating system and they're compatible there. We work to computationally secure and, of course, protect our software and hardware surfaces and interfaces. And we're working to ensure that the input reagents are also screened as best as technology allows. And so I think importantly at this stage, we're really thinking about always iterating and always looking for better ways to improve. And so that's why we feel like these types of discussions and these forums are really critical for the growth and development of cloud labs. So with that, thank you guys very much, and looking forward to the panel and questions.
Great. Thanks so much, Peter, for a really fantastic presentation. It was very rich and shows some of the amazing capabilities that you and your team at Stratios have been able to develop and is happening across the cloud lab industry. I may take a moment to try to explain to our audience who may not be as technically deeply trained as some of our panelists, just to sort of explain the significance of what you're talking about. And Peter, please feel free to chime in if there's anything you want to correct about my paraphrasing here. But what Peter talked about was experiments with proteins and assays to test for the presence of DNA. Those are two different experiments he was talking about. And that's really fundamental to biological research. If you think about cells as a sort of factory floor, the proteins are the machines, they're the things that do stuff. And so understanding the fundamental properties of how protein works is really fundamental to characterizing functions of cells and life as we know it. And so one of the experiments that Peter was describing was that the PCR assay that Peter was sort of demoing at the end is also a very commonly used genetic marker test. And as Peter mentioned, is one of the diagnostic tools that is being used to test samples for the presence of COVID. And this is, and what Peter has shown is really revolutionary because it shows what Stratios and other companies like Stratios are doing is taking work that used to be manual. Grad students, like all of us used to be grad students, we did this manually with pipettes. Used to take weeks, months, many people, and they're basically designing a system where this can be automated, and you can use a computer interface to control robots that will do this at scale, do it faster, enable you to really accelerate progress. And so it's really revolutionary, has tremendous potential, and so thrilled to see the progress that you guys are making. So thanks so much for sharing that, Peter. And obviously it has tremendous potential for driving advances in the future. If I may, so we have about 20 minutes for Q&A. And so we're going to shift gears now to some discussion, and we certainly welcome questions from the audience. You feel free to submit them in the chat. But I'll start off with a question to both Peter and Todd, which is about risk. Both you and your colleagues have clearly done some really great work to improve biosecurity for cloud labs and for the DIY lab community, respectively. Can you share your perspective on some of the potential risks that you are trying to protect against, and perhaps also share your thoughts about how those risks might evolve over time?
So I guess I'll jump in first. So I think it's an interesting and hard question to try to sort of kind of predict where things may go. I think what the DIY community has done successfully so far is at a very basic and really simple level, but that can be more complicated than it sounds, is by building relationships between sort of the sort of safety and security communities and the DIY bio community to both understand what those concerns might be, and then also how to potentially mitigate them. So just as a quick example, from the very beginnings in the US, the DIY community had a working relationship with the FBI, the Federal Bureau of Investigation, which on the surface you would say, that's insane. How would that ever work with a bunch of hackers linking up with a bunch of FBI agents? But what came out of that was a better understanding of both worlds. One, of the FBI knowing what, as a very simple thing, what a community biolab looks like, which is very different than, say, a chemical lab that might be producing, say, methamphetamines. And on the reverse side, the DIY community understood what it was that the FBI not just did, but what they were concerned about to try to be able to sort of mitigate any of those concerns. And over the years, that relationship has been really productive, both for the community labs who can reach out to their local agent if there's someone kind of enters that seems a little off. And we also lean on the FBI as a sort of screen that's basically as a supporter, right? So when we get questions from the press or even regulators who might be concerned, who don't know much about the community, we can use the FBI as a partner to come in and basically say, look, we work with them, they're doing things that we, safely and securely. And so those partnerships are critically important to have, which I think is really the basis of any good governance system moving forward, as clearly things are getting more sophisticated, which we've seen in the community lab space back, if you look at what was going on in 2010 to 9, it's very, very different.
Great, thanks, Todd. Peter, would you like to share your thoughts?
Yeah, I think about a lot of those risks as basically one, in terms of accessibility, and two is the action at a distance, kind of those two issues. And, of course, accessibility is, I think what we all think about is, right, like what we would like to do, of course, is increase access so that, right, science can be itself a democratizing factor. And so, what's the balance of that? And what is the trade-off for who has access to to these high leverage tools, and how do we work with that? And so for us right now, of course, we are working with the trusted partners, and we do that type of validation. And I think that is one of the nice parts, or I think sort of side effects of having sort of that centralized behavior, having that cloud lab is a more centralizing aspect. I think the other side is action at a distance, right? Thinking about what are the additional things that happen in terms of doing science at a distance, of programming science, at your computer and having it happen, in a work cell 2,000 miles away or so. And right now we take a lot of care to work with people to onboard their specific protocols onto our system. And I think as that matures, we'll need to start thinking about things like more maybe machine learning approaches or ways to topologically analyze protocols themselves, and maybe do things that are similar to kind of like first pass of what the Google Play Store does for apps, to ensure that apps are safe there, and then have like a maybe second pass, like a manual review or something for those flagged apps and things like that. So those are all kind of future directions and things that we're thinking about.
Fantastic, thank you both. So if I may, dig a little deeper here, Peter, for you, you and your colleagues at Stratios have taken a pretty comprehensive approach to making sure your systems are safe and secure from a number of different angles, including from the process angle, which you just mentioned, but also wanted to ask a little bit about your sort of how you choose your trusted partners and who has access to your services and how that might change over time. So my understanding from talking to you is that right now you have a small group of trusted partners, you know everyone you work with. And so just wanted to hear a little bit about how you choose your customers. And if you think over time that might change, is the market going to expand? Will there be a future situation where either you or your peers in the Cloud Lab community are needing to screen large numbers of customers and where you don't necessarily know them as well? And how are you guys thinking about that future?
Yeah, that's a great question. And yes, I'll confirm that right now we are working with trusted partners, and they're basically at the threshold where we can manually validate everyone in terms of commercial development and business development. So we have several conversations with all of our users and basically explore what they're working on and what they're trying to do and what their goals are. Even before they're on the system, and we make sure that they're basically validated commercial entity, we basically are working within I guess like a local network in that sense, in the virtual sense. I do think in the future, as you bring up, like the accessibility overall, cloud labs in general will increase, right, and the people who I think use these resources will increase, and some of the things that we've talked about in just the recent past is like just what does an interface look like between Cloud Labs and DIY Bio, and like there would likely be people in the DIY Bio community who would like to leverage these types of tools and things like that as well. And so I think there are probably still going to be good community-driven efforts to be able to identify players that are adopting good practices. I think what Todd brought up in his presentation on sort of having kind of rating agencies be able to certify certain labs. I'm sure that will be a help, and that will be part of that process. I think, as I was alluding to, we're going to need to think about sort of technological solutions too. Are there good ways to topologically analyze protocols and sort of see what's sort of permissive or restrictive, and that can be similar to scanning through a DNA sequence and making sure that there aren't any pathogenic or hazardous sequences in that sequence that's being synthesized or being used or something like that. But I think there will be natural extensions there, and again, I think that's also why these types of dialogues in this community and in this forum is so important to us, that we want to keep iterating on this process as the opportunities, as the scope changes.
Thanks so much, Peter. So I have a question for both of you now about sort of uniformity or lack thereof of best practices across your respective communities. So certainly, Peter has described a really thoughtful approach to security. And so, what's an open question for that community is how uniform is it across the Cloud Lab community? And do we know, to what extent do we even know that other companies are not doing that? What are the best practices? Is that clear? And then, likewise for the DIY community, Todd, you guys have done a ton of work among community labs, promoting best practices and all the things that go with that. But there it shades into other parts of the community, including the Garage Lab community, which is not exactly the same. And so would welcome your thoughts on sort of the distinctions between those communities and how you navigate, how that affects uniformity of practice or sort of creates fault lines.
Yeah, I'll jump in on the Cloud Lab side first and say I think Cloud Labs themselves are a pretty nascent industry, and so there's no great bright line for who's in the Cloud Lab community and who's outside of that for now, and so I think that kind of can muddy the issue a little. But I do think overall that the people, and I would say or colleagues who are also working in this field, the ones we've talked to, are also being very intentional and very conscientious about how they're developing out technologies. And I think that initial process of going through trusted partners and kind of having those conversations and really delving on that side, that is helping, that is happening across the industry at the moment. And I do think, again, we talk about that transition and what it looks like, and I think we all have to be thinking about it and preparing for it.
Yeah, I mean, I would say sort of it's very similar in the DIY community in terms of like, who's in, who's out, like what's considered a community lab versus a garage lab. Are you in the community? Are you not in the community? And that I don't think we can necessarily answer with any clear distinction. I think what's been interesting is that one of the, maybe it's a growing pain, so to speak, is that, when we did those first codes of conduct back in 2011, we knew everyone, right? Because the community was very, at that point, still very small, that it was in a way manageable, where you could get input and develop these kinds of codes of conduct and community practices that was quite easy for people to sort of buy into. As the community has grown, where we now have like a global community bio summit who are trying to figure out what the next stage of that looks like, where you now have a much larger community spread across multiple places with different types of facilities. How do you sort of adapt that community of practice for people to sort of buy into that? And so what are sort of one of our ideas of that, with this, training program on biosafety, we're going to be releasing a biosafety handbook that is specifically designed for community labs and garage labs, because there's differences there, and there's going to need to be some flexible room there in terms of what the rules are compared to sort of a institutionalized lab versus a community lab. And so our hope is that those things kind of trickle throughout the broad swath of types of non-traditional, we'll say, or non-institutionalized spaces, because it's going to be really difficult, I think, to get some kind of universal code of practice to capture everything that's going on. Because while we can sort of know who the community labs are, it's only because they've raised their hand and said, we're here, here's what we're doing. So, how many garage labs there are, I don't think anyone really knows unless they've sort of joined into some of these community aspects and community practices that, have been put in place.
Right. Thanks, Todd. Yeah. So, I mean, this is a really fundamental challenge. How do you secure a space where you don't even have visibility on a number of key actors in the space, and you don't know what fraction of this space is actually not visible? And it's not to say that a part of the space that's not visible is necessarily engaged in activities that are not well intentioned, but you can't confirm that because you can't see it. And so that's it's challenging. It's a challenge that we have to consider, finding effective ways to manage while still securing the access, striking the balance that Peter was talking about a moment ago. And what's really interesting to see in both of these communities is you are already starting to, you're at various stages of developing norms, right? Like what is responsible practice? What is best practice? What is really the best way to, ensure that your work is safe and secure and responsible? And at some point, at least in my view, and a number of my colleagues, it's also helpful to make those more rigorous and find ways to sort of act on those norms and operationalize them more. And so I think it's very interesting that you guys have been thinking about a code of conduct or a seal of approval within your DIY lab community. And from hearing Peter's comments, it sounds like it perhaps could be useful to companies like Stratios to figure out who to partner with, who to trust as customers that are reliable. And so can you talk a little bit more about this seal of approval concept, or this, however you're describing it, the certification?
Yeah, so I would say it's an idea and there are multiple views in the community about whether that's a good idea or not. So that certificate I showed you from that one lab comes from the government agency in Australia that regulates genetic engineering. And they're required to have that in order to actually do any types of genetic engineering experiments. Right? It's a similar type of approval or license that you would need for instance, in Europe, right? So any lab, including a college lab or a startup, would have to have that certificate to legally be doing genetic engineering. So it's based on the actual sort of experiment, right? So if that lab, for instance, in Melbourne wasn't doing genetic engineering, was just doing other biological experiments, they wouldn't necessarily have to have that. That being said, we've been exploring sort of this idea of some type of, like, you know, whether you call it a certification or a seal of approval from the sort of biosafety and security aspect that, you know, a lab could sort of have on hand to sort of show to, say, a funder or show to a company like Peters, or even a company that might be producing their sequences for them, to sort of show that there was some sort of baseline sort of understanding of the practices that they're doing inside those facilities that in essence is making people comfortable enough that they can participate in that community. And that is it's an active conversation. You know, there are pros and cons to that, I think, but I think overall, you know, the labs are already doing most of that would be associated with that. And so it's then a question of, I think, who gives the seal of approval, like who, you know, who runs that, like, do you have to pay for it, like, does it, all of those sort of, you know, administrative types of, you know, sort of questions would have to be answered. And so it's an open dialogue that the community is currently having.
Great, thanks, Todd. And I think, you know, it's great that you guys are having that dialogue. I think that is potentially a replicable model and could be useful across different parts of different communities that are engaged in advanced bioscience research and development. And I want to go to an audience question, which is just really about, well, how do we think about this globally? How can we make sure that all, you know, that cloud labs and DIY labs and all the other systems that we've been talking about today, that they're secure not only locally, but it works globally. And because fundamentally, you know, we're all interconnected, and an accident or an attack in one place can influence, you know, local communities, and it can spread. And so we really have, you know, the concept that we all have a mutual responsibility to protect each other and our global population. And so there are questions about, you know, I think it has two parts, right? Sort of how do you guys think about sort of more global or internationally viable or acceptable norms that could apply for either cloud labs or the DIY community? Can you imagine a vision like that? Would that be helpful? And then also, I think, you know, there's the question of, you know, and I think there's the question of institutional home for that. What's the right home for that? And perhaps I can comment on that. I think I'll just note that there is not currently an institutional home for that kind of work. So if Todd wanted to do a seal of approval or a certification, there's nowhere that can live. They would have to build that. And that's something that we are, you know, at NTI are asking ourselves, how do we promote global governance in a way that can protect everyone and sort of glue all these things together into something that's more integrated? But yeah, I would welcome your thoughts, both from you and you, Todd, and you, Peter, about how does that sound feasible to you? Would that be helpful to you? What do you think are some of the benefits and challenges of that kind of approach?
So I can start. So let me frame this maybe in my own experiences. So I do a lot of work in a different UN agency, which is the Convention on Biological Diversity, around synthetic biology and gene drives. And so in my mind, trying to sort of develop a global framework like that in those types of systems is going to be difficult, if not impossible. For one, just by the pace at how those institutions operate. Now, I think they do extraordinary work, and there's some really great, important things that those types of systems can do. But for, I think, communities like this that are rapidly changing, rapidly expanding, it'll be really difficult to work within that kind of system unless they somehow change how they operate. And so we've been, you know, tackling this from like I said, from a bottom up, building those relationships kinds of models where, you know, like the work that Piers had talked about through IGEM, right? Where they have, in a sense, built a global sort of network and community of practices within their community. And a lot of that work was done, again, by building relationships one inside the community, but then expanding outward to get buy-in from individual countries or localities that sort of can better understand what's going on. And so for me, I think the way to do this is to figure out how to build those relationships and not focus so much on kind of some kind of like overarching organizational structure, because I'm not sure those are set up at the moment to handle this kind of distributed technologies.
Thanks, Todd. Peter, did you want to chime in?
Yeah, I'll echo too. I mean, I was really thinking about something similar with both of these initiatives, right, both DIY Bio and Cloud Labs, that it is something where perhaps that organic and that bottom-up approach will just kind of naturally work, right? I think about it from the Cloud Lab side of as it expands out to different countries and sort of think about the different rules and regulations therein, because it's kind of has this centralizing aspect of it that right, it's all managed on the same software systems. It's all able to basically use the same same interface, same devices. That it becomes a process of basically taking what happens on the regulatory side as like I guess maybe the most stringent or finding what's the compatible regulatory things and just incorporating those and kind of distributing them through the network, right? So I think there can be kind of that principle, and probably the same in the DIY bio community, maybe from a more organic sense, like a people-driven sense. That yeah, I think what Todd was saying is that as you incorporate groups internationally, then you get to see what the best practices are throughout the world, right? And then you can like funnel that up through the DIY bio community or through different sectors of the DIY bio community, and have groups that are all following the best practices that have been developed. So probably maybe kind of analogs there.
Great, thanks both of you. Yeah, I wanted to sort of respond to that really quickly. It makes a lot of sense. I mean, I think as we've all noted today, the rapid pace of change is such that government and conventional institutional structures cannot keep pace, as you both noted. Absolutely true. And I think, I think the benefit of a global approach is, very much an extension of what you guys are talking about with this organic, fast-moving, innovative, bottom-up approach. And I think the question is just how do we make sure that we can put all that together and ensure that there is like maximum uniformity globally to the extent that's necessary for really robust safety and security, and make sure there aren't big gaps. But maintaining that sort of innovative, moving quickly, bottom-up energy, and not sort of bogging it down with sort of slowing it down with institutional structures that can't keep up. So there's a real tension there, and I think there's more work to be done in figuring out how to strike that balance. So I just want to give each of you really quickly an opportunity to share really like 30 seconds, last word, what your last point that you would like our audience to take away from this conversation, and then I will summarize key takeaways. Todd.
Yeah, I guess my key point is that we want to work with partners, and so reach out to your local community lab if there's one in your country or in your locality, and build those relationships and partnerships to try to actually sort of expand out these communities of practices. It really is a global community, and so those partnerships really need to be there. So I think it's critical that the biosecurity community has a better engagement process with your local labs or the global community as itself. So that would be my take home. To please reach out to us. We would love to sort of partner with you to learn from you, and you can learn from us, so we can work through these issues as they evolve. Thanks, Peter.
Yeah, there is a lot of innovation and I guess ingenuity happening in the cloud sphere, and we're really pushing to accelerate science and scientific discovery. And I think within that, we want to keep iterating on this conversation on how we can get to better and better practices and like best practices in the end. And I think that whole balance, right, that Jamie was talking about, is about establishing those relationships and keeping up the communication. So I also encourage that we are active participants and want to continue to be active participants within this biosecurity sector. I think just the mutual understanding and getting to how do we develop technologies that will meet biosecurity needs is deeply important to us. So thank you very much for that.
Fantastic. Thanks to you both for excellent presentations and discussion. I'll just take a quick minute or two to summarize key takeaways. So first, cloud labs, by automating aspects of life science research, have the potential to fundamentally change the way that we do biology, with the potential for tremendous benefit and innovation. The DIY bio community is an enabling citizen science across the US and around the world. Also democratizing access to science and presenting excellent opportunities also for innovation. Both of these communities have worked proactively to build in processes and best practices to ensure that the work is safe and secure. But we have more work to do. We still need to guard against exploitation by malicious actors who may seek to cause harm or accidents. And as one of the questions pointed out, this is really a global issue. And we have to find a global set of solutions, navigating the challenges of sort of bridging between fast-moving, innovative bottom-up approaches, which are critical, with some sort of globally viable solution where we can ensure that we don't have huge gaps in implementation. I really like what Peter said about the importance of ongoing partnership, and I think we're really lucky to have people like Peter and Todd and others in the community that continue to engage in dialogue and partnership with people in the biosecurity community, including within the UN system. And it's going to require that. We're going to require ongoing efforts by governments, academia, private sector, and the international community to really bolster biosecurity and biosafety around the world to address emerging biological risks that come with advances in technology, and including a global approach. So with that, wanted to thank the panelists again. Thank all of you for your attention. And for the opportunity to share these thoughts with you today. And I'll turn it back to you, Giacomo. Thank you very much.
Thank you, Jamie. Thank you, Todd, and thank you, Peter, for an incredibly engaging and interesting panel. I've learned a lot on things that are, from an outsider perspective, can be sometimes quite impenetrable, but because of your very clear presentation and discussions, you really helped us understand better what does DIY, bio community stand for? What are cloud labs and how do they work? I think it was a very interesting panel. So we are approaching the end of the first day of the Innovations Dialogue. And in this first day, we really got a sense of how new technologies, new processes, and new actors are shaping the present and future of innovations in the life sciences. It's really not only a technology-driven innovation, but because of technology-driven innovation, we've heard how new actors are entering this arena. New processes have been developed, and kind of a common thread across all of the contributions that we heard today from our speakers, starting from Andrew and through the two panels, is that in a way this innovation requires new types of instruments and frameworks, and whether they be in governance or regulation or simply new partnerships, new tools, new partnerships, and new frameworks. Now, this paves the way very well to our second day tomorrow. Tomorrow, we're going to have a last panel exploring technology, particularly looking at delivery and nanobiotechnologies and how they're used for delivery of biological agents. But then we're going to move, as I said at the very beginning, to the second kind of thematic part of the conference, which is exactly going to try to answer questions around governance issues. What are the gaps? Are there gaps? If yes, what are they? And how can they be filled? Do we need new instruments, or can existing instruments be adapted and used to fill those gaps? And then the last panel of the Innovations Dialogue will try to bring this back to the multilateral level to explore, in all of this, given everything that we've heard and learned in the last 2 days, what is the role of the UN and of multilateral architectures to deal with these issues. So I really hope that you enjoyed this first day of the Innovations Dialogue, and I'm really grateful for that and for all the support team operating behind the scenes. You will have seen in the chat box a link being shared to our feedback form. I really encourage you to visit the link, or to scan the QR code that you see on the screen with your mobile. This will take you to our event feedback form, and we really value your feedback. We always try to improve our events, and your contribution to that will be much appreciated. With this, I wish you a great evening, and I hope to see you back online tomorrow. We will make a start at 9:30 Central European Time, and I hope to see you tomorrow. Thank you, and have a great evening.